Good afternoon, good evening, everyone, depending on the places where you are in. Welcome to the 11th webinar of ONWARD. ONWARD, as you all know, Open Network for Water-Related Diseases. It's a project funded by the GCRF Global Challenges Research Fund, UK. It's led by Dr. Milton Campbell and Dr. Shubha Satyendranath. And the network is dedicated to developing a network of people, whether they be scientists, stakeholders, or laymen, sharing a common interest in better understanding of environmental controls on outbreaks of water-related diseases. So today we have Professor Archie Clements with us. Professor Clements is a veterinarian and infectious disease epidemiologist with PhD from the Royal Veterinary College, University of London. He has an equally impressive career record spanning more than 15 years, holding senior academic leadership positions in higher education and health service settings. He was Pro-Vice-Chancellor of Curtin University, Western Australia, and Director of the Research School of Population Health at the Australian National University. At present, Professor Clements is the Deputy Vice-Chancellor, Research and Innovation at the University of Plymouth, also holding the position of Professor of Global Health in the Peninsula Medical School. Professor Clements' research expertise is on the spatial epidemiology of parasitic, bacterial, and viral diseases, predominantly in tropical regions of the world. He has also worked on community-based intervention studies, exploring the role of multi-component integrated strategies for sustainable infectious disease control. So we have a world-renowned health expert who has approximately 300 peer-reviewed publications and has handled projects worth £18 million as chief investigator. Today, Professor Clements will speak on using spatial models to understand the epidemiological drivers of waterborne diseases and to produce maps useful for disease control. Welcome, Professor Clements, to the ONWARD platform, and the floor is yours.
Thank you so much for that lovely introduction. It's really humbling to be invited to speak with you and to and hopefully to meet you all. So yeah, as Nandini mentioned, my presentation is on the spatial models and how they can be used to help understand the epidemiological drivers of waterborne diseases. But most importantly for me, most of my research has been integrated within the health service delivery environment, and I'm interested in producing maps that can be useful tools for disease control. Just checking, have I got this? Have I shared my screen properly so that you're all seeing the slide in presentation format?
Yes, we are seeing.
Excellent. Okay. All right. So anyone who's worked in epidemiology or in infectious diseases, particularly those of you who worked in waterborne diseases, will know all about John Snow, who was a physician and an epidemiologist in England in the 19th century, who was interested in the notion that cholera, which was a disease that killed lots and lots of people and caused intestinal disease that led to death, was not transmitted as people thought at the time via miasma or foul-smelling air, but was transmitted via the fecal-oral route, with water being the main mechanism. So he undertook a study of a cholera outbreak in London in the mid-19th century, and he mapped the location of cholera cases in London and demonstrated that they were clustered around pumps that attached to wells that were managed by a particular company within London. And here we can see one of those maps that he produced. So the little dots are the cholera cases and the black squares are the pump sites. And you can see that they're clustered around a particular pump in Broad Street, London. And he convinced the authorities to remove the handles from the wells that were managed by this company. And that coincided with the end of the cholera epidemic. although arguably it was already in decline because people were so terrified by cholera in London at the time that they were all leaving the area. So that is fundamentally what we're still doing today when it comes to spatial epidemiology, particularly in an operational context, using maps to give us insight into the drivers of disease, and then also using them as tools to inform public health decision making that then hopefully have an impact on control. So moving forward, this is moving into the 21st century. This is some work that I did with some colleagues in Kathmandu, and this is looking at another enteric gram-negative bacteria, Salmonella typhi, and also Salmonella paratyphi, so typhoid and paratyphoid fever in Kathmandu. And what we did is we, like John Snow, we mapped the location of cases. These are people that had come into the hospital, which is located in the middle of the study area here, back to their household address, and we typed them. So we identified them as either being Salmonella typhi or Salmonella paratyphi. So we had some advantages over John Snow. We had genomic information on the pathogens. In fact, we knew that the species of pathogen at the time that John Snow did his work. They didn't even know that that cholera was caused by a bacteria, Vibrio cholerae. We now have genomic tools. We can type these pathogens. We can identify individual transmission events. We can look at particular clusters of particular genotypes to identify specifically important environmental exposure points. The other thing that we were able to do in this study was incorporate the distribution of a population at risk. So you'll remember from the map that I showed you from John Snow's map, it was just the cases, but it didn't show risk because it didn't incorporate the distribution of the underlying population. It might well be that those cases were clustered around the Broad Street pump because that's where all the people lived, and that may have reflected the distribution of the population rather than the distribution of disease risk. So here, we've undertaken a spatial case control study where we've used information on the cases, but we've also mapped the density of the catchment area for the hospital and then related the density of cases to the density of the catchment area to produce these maps on the right, which are risk maps. They're showing hotspots of transmission. And so the map at the top there is a very well-defined spatial cluster of Salmonella typhi, and then the map at the bottom is a slightly more diffuse cluster of Salmonella paratyphi. And interestingly, like John Snow's map, these hotspots are actually linked to a particular well near the river in Kathmandu. And then so these maps are useful tools then to sort of plan where interventions need to be undertaken within Kathmandu city. So aquatic environments are really important when it comes to many infectious diseases. So a large number of infectious diseases are modulated by the aquatic environment, and there's a very high global disease burden associated with waterborne infections. So water can be a physical vector, so as is the case with Salmonella typhi and cholera. It's a physical vector in that water can move through an area, it can collect fecal material, it can concentrate fecal material, and then provide an environment where pathogens can survive and then be transmitted onto people, either through physical contact or even through drinking the water. Water also provides an essential habitat for free-living bacteria, as is the case with Leptospirosis, which can either exist in a free-living form in water or in soil. Water can also provide an essential habitat for free-living stages of parasitic diseases and their intermediate hosts. So I've listed a couple of diseases here, really interesting diseases with fascinating natural histories. So one is Ophisthorchis viverrini, which is the cause of a fluke infection in Asia. And it's a really interesting disease in that it's got two intermediate hosts, both of which require an aquatic habitat. So a snail host and a fish host. Another one is Schistosoma species. So there's Schistosoma japonicum in Asia and the Schistosoma species in Africa that have a snail intermediate host, and that's an aquatic snail. And so the aquatic environment is essential for the existence of this, of the life cycle of this pathogen. And then there's another one called fasciola, which is a really important disease both in livestock farming and also in humans, that has an aquatic snail as its intermediate host. Water can also provide concentration points for reservoir hosts. So if we look at Schistosoma japonicum, a major reservoir host in Asia is buffalo or the carabao. And these buffalo are attracted to water. They spend part of their life in water. They defecate in water, and they, because they're much bigger animals than humans, they are a major driver of the transmission cycle. They produce a lot of parasite eggs that then go into the water when they defecate, and then those parasites infect snails, and then the life cycle continues. Also, leptospirosis in water provides important concentration points for livestock and rodents who transmit onto humans. Very importantly, in terms of global disease burden, water provides habitats for developmental stages of arthropod vectors. So here are a couple of very well-known ones: malaria, which is transmitted by Anopheles mosquitoes, and dengue, which is transmitted by Aedes mosquitoes. These mosquitoes require aquatic habitats, and therefore the existence of water bodies is important in the transmission of these globally important infectious diseases. And there are many, many other examples of arthropod vector transmitted diseases that include parasites and bacteria and viruses. Water also provides a critical environmental contact point for humans. So that could include an occupational contact point through fishing or through paddy farming, or it could be an important contact point for people undertaking domestic activities like washing clothes, washing children, washing household utensils. And also importantly, water can provide a critical contact point as part of recreational exposure. So that might be people whitewater rafting or swimming or undertaking other recreational activities that involve water. And really importantly, use and misuse of water as a component of hygiene and human waste disposal is an important environmental risk factor. So if we think about hygiene, particularly in developing parts of the world, it requires the existence of a water source in order to wash or to flush latrines. And so the presence or absence of water can be really important in terms of the quality of hygiene that is achievable. If we think about trachoma, for example, in Africa, the availability of water to wash the face is a really important determinant of risk. If we think about soil transmitted helminths throughout the tropical regions of the world, we need water in order to reticulated water that can lead to the development of functioning latrine systems that are essential for sewage disposal. Because if we don't have that, then people defecate in the open, and that can result in the transmission of soil transmitted helminths. And then in terms of the misuse, people defecating in water is a major cause of the transmission of infectious disease. So schistosomiasis, Ophisthorchis viverrini infections would not exist if people didn't defecate in water or urinate in water and allow the completion of the parasite life cycle. So we need to undergo a lot of health education in order to prevent that misuse of water to prevent disease. And there are many other examples, people allowing animal feces to run off into water sources that can then lead to the promulgation of antimicrobial resistant bacterial infections that then can end up in infecting humans. So water is very important for infectious diseases in many, many different ways. So some of those interactions can be very complex. So here I'm showing a life cycle for leptospirosis, which is a very complex bacterial disease that has a multitude of different kinds of reservoir hosts. Can be sylvatic cycles with rodents and wildlife, can be more domestic cycles with livestock or companion animals. The leptospires are excreted in the urine of these animals that can end up in the soil or the water, and that can then end up infecting humans. Here is a spatial epidemiological study that we did on leptospirosis in American Samoa in the South Pacific. We undertook a survey of people on Tutuila, which is the main island of American Samoa, and we did a sero study, and we determined whether or not people were positive or negative to leptospirosis. And here we have a map showing the distribution of the seropositive cases, which are the red ones, and the seronegative individuals, which are the white ones. We did a spatial cluster detection analysis using Kuldorf's scan statistic, and we found a statistically significant cluster of leptospirosis cases here in the middle of the island. We were interested in understanding that further, and so we built a spatial model that incorporated a number of covariates with the purpose of spatially predicting the risk of leptospirosis in Tutuila Island. So the map on the left is vegetation, the map on the right is a digital elevation model. And it's quite interesting, you can see this really helps you visualize the distribution of the population of Tutuila. We can see that most people in Tutuila live near the coast in the low-lying areas, but there is a cluster of settlements up here on the top of a hill. And this is actually that spatial cluster of infection that I showed you in the previous slide. And from that spatial prediction model, we were able to predict or estimate risk across the inhabited areas of Tutuila. And so this is the predicted seroprevalence of leptospirosis on the island. And this is the spatial cluster that we detected using the scan statistic. And we also predicted using our spatial prediction model that this would be an area of high predicted risk. This cluster up here is associated with a genotype of leptospira that is associated with a sylvatic transmission cycle that has sylvatic bats and rodents as the main reservoir. And so this analysis demonstrated that that was a particular transmission cycle with a particular set of reservoir hosts that needed to be addressed through public health measures on the island. I mentioned malaria. Malaria is a fascinating disease of great global importance. It has a very complex life cycle. Part of that parasite life cycle exists within the human host, but part of it exists within a mosquito host. And I think one of the critical things that makes many diseases really sensitive to environmental conditions is the existence of a part of the life cycle that exists outside of the human host, whether that be as a free-living organism, as is the case with leptospirosis or Salmonella, or soil-transmitted helminths, at least parts of that life cycle, or as an organism that's transmitted via a vector, which in itself is very sensitive to the environment. Those conditions mean that the environment then becomes very important. So if a parasite or a virus or a bacteria is transmitted by a vector, the fact that that vector is sensitive to environmental conditions means that disease exists only in areas that have particular environmental characteristics. And again, if there's a free living stage outside of the human host, the climate or the microclimate or the physical environment that determines whether or not that free living stage can survive will then determine the spatial distribution of that disease. And those principles are fundamental then to our ability to predict the distribution of the disease on the basis of environmental variables or environmental characteristics. So here we can see that the mosquito depends on the existence of aquatic habitats to enable the completion of its life cycle. So the mosquito lays eggs in an aquatic habitat, the eggs hatch into larvae, which then mature into pupa, and then an adult emerges, and then that adult then needs to go on and have human blood meal, or that in order to survive. And it's when that mosquito takes that blood meal that it picks up the infectious stages of the parasite. Those parasites then go through various stages of maturation in the mosquito, and then the mosquito, when it takes the next blood meal, then infects the next person. But essential to all of that is the existence of an aquatic habitat for that mosquito population to propagate. But not all malaria mosquitoes are the same. So there are a wide range of different species of Anopheles that transmit malaria, and each of those species has a very different set of environmental requirements. And I'm going to draw your attention to the purple ones here in Melanesia and the ones up here in Afghanistan, because I'm going to show you two examples where we did spatial prediction for malaria and using very different information about the aquatic environment in order to predict malaria risk. So the first one is Afghanistan. So this is data from an epidemiological study of Plasmodium vivax malaria in Afghanistan. And the red dots are places where, communities where people had malaria with various different levels of prevalence. And the green dots are the communities where people were not found to have malaria. And I built a spatial prediction model using different covariates. So this one at the top is the one that you'll be most interested in as people who care about waterborne disease. This is distance to a perennial water body, or which in Afghanistan is mostly rivers and streams. And then the second covariate is altitude, meters above sea level. And so we built a multivariate spatial prediction model that looked at the relationship between disease prevalence and each of these covariates simultaneously, and then used the relationships from that model to predict the distribution of risk of Plasmodium vivax infection in Afghanistan. We can see that we predicted areas of high risk in the east of the country, which is sort of lower lying areas, and in the river valleys that go up towards the highlands of Afghanistan around the rest of the country. And these are areas of the river valleys are important because that's where the mosquitoes that transmit malaria are. Now here we're looking at New Guinea, which is very different, Papua New Guinea. Again, we did a survey, but the variable of interest here with respect to aquatic environments is distance from the coastline. And that's because in New Guinea, the malaria transmitting mosquito, the predominant one is Anopheles peralti, which thrives in brackish environments close to the coast. And so distance to coastline was an important predictor, recognizing that the mosquitoes were more prevalent in these brackish aquatic environments along the shore. And so here we were able to predict the spatial distribution of this is Plasmodium falciparum malaria in New Guinea using these different spatial predictors: rainfall, temperature, and distance to coastline. Here's another spatial prediction study for this is Plasmodium vivax malaria in Tanna Island in Vanuatu, which is another country in Melanesia. And again, we had distance to coastline as a covariate, and we predicted a higher risk of infection in coastal areas because, again, that's where the mosquitoes were present. Interestingly, Plasmodium vivax and Plasmodium falciparum, which are the two major species of malaria in the world, are transmitted by the same mosquitoes, Anopheles peralti in Melanesia, and hence you get this coastal distribution of the disease. There are some other elements of this image which I want you to bear in mind. One is the 95% confidence intervals of our predicted risk on the right, and the other is the semi-variogram, which is the spatial component of the model, which I'm going to talk about later. I also talked about Ophisthorchis, and it is really a fascinating disease. So this is the life cycle of Ophisthorchis. Ophisthorchis is caused by a fluke parasite, Ophisthorchis viverrini, which lives in the Mekong subregion of Southeast Asia. And the adult is this fluke that lives in the biliary duct, the bile duct of the liver. And it causes fibrosis and inflammation and secretes carcinogens that can result in liver cancer. And so as a result, particularly in northeast Thailand, you see the highest rates of liver cancer in the world related to the to chronic infection with this parasite. So it lives in the bile duct, it excretes eggs in the feces, and those eggs then go in, if they come into contact with water, will hatch, and then immature stages will then invade snails if they're present in that water body. There are various stages of maturation of the parasite within the snail. Then once the parasite's matured in the snail, you get free swimming cercaria that are released from the snail that then invade a fish. And then those cercariae form cysts in the fish. And then if the humans eat raw fish infected with these metacercariae, they then become infected. And then those stages of the parasite then mature in the gut, and then they go into the bile duct as adult fluke. So there are multiple intervention points in this life cycle. So you can intervene between the fish and the human by health education, telling people not to eat raw fish. Unfortunately, raw fish are a major food source and a cultural delicacy in northeast Thailand. People love eating raw fish in northeast Thailand. And so it's a challenging task to stop people from doing that in order to prevent the infection. You can also prevent people from defecating in water using latrines. There are various environmental interventions you can do, for example, mollusciciding, which can be quite damaging to the environment potentially, but you need to use an integrated approach there. So moving on to some spatial epidemiological approaches. Here we have a study area around the Lower Lake in Khon Kaen region of northeast Thailand. Again, we did a survey. So the little purple triangles are the communities that were sampled. Here is the prevalence. This map in the middle is the prevalence of Ophisthorchis viverrini monoinfection. We were also interested in co-infections with another parasite, Strongyloides stercoralis. So that's prevalence of that parasite, and then prevalence of co-infection. And as for the other studies, built a spatial model and then did spatial prediction showing the areas of high prevalence, high risk of Ophisthorchis viverrini monoinfection and Ophisthorchis viverrini Strongyloides stercoralis co-infection. And so these are the areas that then need to be prioritized when it comes to health education, deworming, distribution, mass drug administration, and other forms of environmental modification that prevent the disease. We also did some research looking at the spatial co-distribution of periductal fibrosis, which is diagnosed using ultrasound, and infection, and we show that there is a significant overlap in the spatial distribution of the chronic liver changes that are precursors to cancer and the infection. Another really interesting disease is Asian schistosomiasis caused by Schistosoma japonicum. I mentioned the carabao, which are the most important reservoir host, but also the aquatic life cycle of the parasite that goes through an intermediate host snail. So again, demonstrating the importance of aquatic environments to the transmission dynamics of this disease, both directly because it provides the habitats for the snail in the free living stages of the parasite, but also indirectly because it provides an important concentration point for the major reservoir host, the carabao. Here's some spatial epidemiological study we did on Schistosoma japonicum in the Philippines, where we did a national survey. And you can see that there's high prevalence in communities in the Visayas and in Mindanao, and in small foci within Luzon, and then building a spatial model predicting environmental risk of Schistosoma japonicum in the country. Some areas where we know schistosomiasis is already highly prevalent, other areas where it's not highly prevalent yet, but which are environmentally suitable for the disease and where we may want to do enhanced surveillance. And in fact, subsequent to this analysis, a new focus of infection did emerge in the northern part of Luzon. Schistosoma hematobium and Schistosoma mansoni, which are the African forms of schistosomiasis, are a little different in that they don't have a major bovine intermediate host, and the aquatic environments are much more important as direct sources of infection because they provide habitats for the intermediate host snails and the free living stages. And I wanted to show you this analysis because it was taking those really interesting spatial prediction studies to the next level and thinking about how we can use this information to actually inform a real-world public health program. So here we have a survey. The data is getting a bit old now, but this is showing prevalence of Schistosoma hematobium and Schistosoma mansoni in the northwest region of Tanzania, just south of Lake Victoria. You can see that Schistosoma hematobium is much more widely distributed than Schistosoma mansoni. Mansoni occurs mainly or almost exclusively along the lakeshore because the snail that transmits Schistosoma mansoni prefers stable aquatic environments that are provided by lakes. So they're stable environments, they're deeper, they're cooler, and they have particular vegetation types that this snail prefers. Whereas Schistosoma hematobium is transmitted by a different snail that prefers irrigation areas, streams, and ditches where water moves and where the qualities of the water are quite different. And so you get a very different spatial distribution of this parasite in more inland areas. So again, I did a spatial prediction model, predicted the risk of these two infections, which is where all of these other studies that I've shown you stopped. But what we did then, which I think is the most important aspect of this study, is we translated these maps into a public health decision-making tool. So these two infections, Schistosoma hematobium and Schistosoma mansoni, are both treated by the same drug, praziquantel. And the WHO recommends that where prevalence of either of these parasites is above 50%, you do annual mass drug administration of the entire community. And so I translated these two maps into a contour map showing areas where prevalence of either of these infections is above 50%. And so these are areas of the map where we need to do annual mass drug administration with praziquantel. In areas where prevalence is between 50% and 10%, you do biannual mass drug administration of school-aged children and pregnant women. So these are the areas between the blue and the green, the blue and the red contours on the map. So you have so you stratify the map by intervention strategy according to the prevalence that you predict from the spatial prediction studies. And then areas outside the blue contour, these are areas where prevalence of either infection is below 10%. You just make drugs available in the rural health centres, and people who are showing signs of infection can then go and get treated. So what this does then is it translates a spatial prediction study into a real world decision-making plan that introduces efficiencies into the control program, because you can focus your resources in the areas where they're most needed, and not allocate resources in the same extensive way in areas where they're not as needed. And so this is really important, particularly in resource-constrained settings where you need to have a strong evidence base in order to focus limited resources. Now, if you remember in the map that I showed you from Vanuatu, there's also this the uncertainty information. So these maps were derived from a modelling approach called model-based geostatistics, which uses a Bayesian framework. And the really good thing about these approaches is that they can give you the spatial predictions are actually distributions. They're uncertainty distributions. And so you can summarize them according to the median, which is the most likely value of risk in a given location. But you can also summarize them according to 95% credible intervals or confidence intervals or prediction intervals, as I call them here. And so the use of this information is that you can say that there are areas of the study area where we can be absolutely certain that disease risk is below a particular threshold. Okay, so that's the bottom map here. So we can confidently exclude the green areas of this map because our because our uncertainty estimates suggest that we're very, very, very certain that there's no disease in those areas. And then there are other areas of the map where we can be absolutely certain that the disease is present at a high level. And so that's the areas in the middle here. And so if we were really constrained in our resources, we would target these areas on the map on the top right, because we're really sure that disease risk is highly prevalent there. If we had lots of resources and we wanted to only exclude areas where we were really certain that the disease was not present, then we could use the bottom map and exclude these green areas. So that's the additional information you get from Bayesian modeling. You get this additional richness of information that you can then match to your decision-making criteria, the availability of resources, and the consequences potentially of either excluding or including a particular area. There are other ways of representing uncertainty. So this is data from West Africa on schistosomiasis. This is showing that the probability of disease or of infection exceeds a particular threshold. So I mentioned that WHO recommends that you do annual mass drug administration everywhere where prevalence is above 50%. So you can look at the map on the right and you can say that there are areas of the map where prevalence is very unlikely to exceed 50%. So those will be the blue areas, and those are areas where we're pretty confident that we don't need to do annual mass drug administration. Whereas in the red and the pink areas, we're very confident that we do need to do it. And then there's the areas of uncertainty in between where we may or may not need to do annual mass drug administration, and we may choose to do it or not, depending on the availability of resources or our desire to collect new information to reduce uncertainty. Another thing that I wanted to mention is spatial sampling. So often as spatial epidemiologists, we're given data that was collected for some purpose other than spatial analysis and spatial prediction. And that data is suboptimal for the purposes of spatial analysis, and it results in areas of the map where you've got higher levels of uncertainty, areas where you've got more certainty because you've got an uneven distribution. I'm referencing here the work of Peter Diggle, who actually developed the concept of model-based geostatistics, and he did a lot of work around optimal spatial sampling designs. I like the map on the left because it's actually showing sampling for measuring salinity in the sea, and it demonstrates that you can do spatial sampling both in terrestrial environments but also in maritime or aquatic environments, depending on what it is that you're wanting to measure. And his research demonstrated that the most optimal sampling approach is one called a grid plus closed pairs design, where you impose a grid on the study area, which has dimensions that ensure that you capture all of the important spatial heterogeneity, but you also have a subset of locations where you sample more densely in order to characterize the left-hand side of the semi-variogram, which is the bit that helps you quantify the scale of spatial correlation. It helps you define the spatial correlation structure of data. And so what I did in West Africa is I actually had the privilege of designing these surveys from scratch, and I was able to use a grid plus closed pairs design in order to provide the most efficient sampling to inform the spatial analysis study. And so if you zone in on this map, you'll see that I've got a really good even spatial coverage of the study area because I used a sampling grid, but there are some areas where I've sampled more densely in order to characterize the spatial correlation structure of the data, which is important for doing spatial modeling. Okay, and the last bit of my talk is the translation of maps not only into policy tools, but also into operational tools. And the framework that I've been working on is the Spatial Decision Support System framework. So what are spatial decision support systems? They're electronic systems that are usually based on a geographical information system that provide operational information that can be useful for decision-making in a disease control setting. So the example I'm going to give here is a spatial decision support system that we developed for malaria elimination in Solomon Islands and Vanuatu. And we rolled out the spatial decision support system in the provinces of Solomon Islands and Vanuatu that were embarking on malaria elimination. And the key operational decisions that were needed to be made in order to support elimination were working out where to intervene, working out how to scale up frontline interventions, which included distribution of bed nets and doing insecticide spray, and doing high resolution surveillance and targeted response. So in terms of geographical reconnaissance, what we did is we mapped and enumerated all households within the elimination provinces using digital handheld devices, which created a geospatial database that could then support the frontline interventions and the surveillance response. And so in our spatial decision support system, that's what this looked like. So the blue dots are all of the households in this part of Solomon Islands, and they're all fully enumerated. We know the number of people who live in each of these households, and they've got an identity. Each of these households has got an identification number. Then, in terms of the scaling up of interventions, we were able to use that enumerated database to provide field personnel who were doing indoor residual spraying and bed net distribution with a map of the households that they needed to visit. So they could go and visit those households, they could record the spraying of household structures and the delivery of bed nets, and then they could produce secondary maps that showed households that had been, where the intervention had been rolled out, and households where the intervention hadn't been rolled out, and then that would enable us to go back and do mopping up exercises to increase coverage in those areas that have been missed. And this is just showing sort of a higher resolution. So here we have clusters of households that had been reached, clusters of households that hadn't been reached, and that was then useful operational information that enabled us to go back and target those households in mopping up activities. And then the third component of the spatial decision support system was surveillance response. So when someone got sick, they would go to a health centre, there would be a rapid diagnostic test, they would be diagnosed with malaria. That information was radioed through to this surveillance response team and captured in the spatial decision support system. So here we have an island in Solomon Islands, and we have the active malaria cases demonstrated with the red stars. And then we could produce zones, investigation zones around those index cases where we would do reactive case detection and attempt to stamp out transmission. So there you go. That's maps from epidemiological exploration to the development of real-world intervention strategies, and then translation into operational tools. That's it for my talk. Looking forward to your questions, and thanks for your attention.
Thank you. Thank you, Dr. Clements.
You're welcome.
So we'll pass on to the questions. We have 4 of them now. And the first one is by Sanusi Mohammed. He has congratulated you for the excellent presentation. And he has also asked, does the study involve use of satellite images and field work?
So some of them, so in terms of satellite images, yes. So for example, I'm just going to show the now how do I do this? No, I can go back, and that's going to take me a while. How do I get out of that? All right, and then if I go there, and I go to the example from New Guinea. Okay. Right, sorry. Well, I'll show this one. So many of these covariates that we use in spatial prediction studies at that sort of national scale are derived from satellite remote sensing. So for example, we often use, where we're interested in proxies of rainfall or proxies of vegetation cover, we might use normalized difference vegetation index, which is derived from satellites measuring the ratio of green and red light on the surface of the Earth. Also, the images that are showing the location of water bodies. So, for example, the streams here and rivers in Afghanistan, they will probably be derived from satellite remote sensing images. we can, if we're interested in rainfall, we can either use satellite remote sensing data, or we can use weather station data, and we can interpolate that weather station data. So the example here for New Guinea shows rainfall at the top right here, and that information is interpolated from weather station data. Whereas the temperature information, again, that'll be interpolated from weather station data. So we use a variety of different sources that will include satellite remote sensing. It'll include weather station information that's then interpolated to cover the entire study area. It may include information that's derived from aerial photography, or even data that's collected on the ground. Could be from digitizing cartographic, digitizing maps, for example. The field work usually involves the collection of the disease data itself. So the map that I'm showing on the top left is data from a malaria survey, where people went to individual communities and assembled people, took blood samples, and then used microscopy or rapid diagnostic tests to identify who was infected with malaria parasites. So we integrate that information on the disease with information on the environment that may be derived from satellite remote sensing in order to predict the risk of disease in non-sampled locations.
Thank you. And the next question is by Pedro. He has also thanked you for the fantastic presentation. What he would like to know is that, have you performed any work on mapping malaria in Angola? Same for schistosomiasis.
No, I haven't done any work in Angola. I've done a lot of work on schistosomiasis in Africa, but not in Angola, that I can remember. I've done a little bit of work in Angola, but not on malaria or schistosomiasis. But certainly Angola is highly endemic for both diseases, and so it's part of the world where more schistosomiasis and malaria research needs to happen. So if you've got any connections in Angola and you want someone to collaborate with, I'm open for business.
Yeah, he has also asked whether these spatial models are freely available to be adapted to other geographies, and what kind of data is needed to map malaria.
So the spatial models themselves are not freely available, because they are, they sort of utilize, they require a lot of steps that involve moving data between databases and software packages, and then intergeographical information systems. There's no automated process to derive these models. And then you need to go through, many detailed steps and to calibrate models, construct them, calibrate them, validate them, and extract the outputs and summarize them and then map them. So they're all manual steps, and which means that these models are not then freely accessible. I mean, the code for the models could be made available. A lot of the papers that I've got in the appendix have got the sort of the code for the Bayesian models, for example, but you can't really run that unless you've actually got the data that feeds into the models. So yeah, that is a downside. I mean, if you look at the spatial decision support system at the back end of my talk, that doesn't actually involve very much sophisticated modeling at all. And the entire purpose of that approach is to make mapping accessible to people who don't have the highly specialized skills that are required to do the spatial modeling. So, but again, there's a lot of complexity there in developing those systems to make sure that they speak to the data collection systems and the resource mobilization systems that exist within countries undertaking disease control. So that's my answer to your first question. Sorry, Nandini, what was the second question?
That was about what is the kind of data that is needed for mapping malaria?
Depends on what kind of mapping you want to do. So if you're wanting to do spatial prediction and risk mapping, then you need data on the disease and you need data on the environment. So the data on the disease that you need is either the prevalence of infection in a location, or the incidence of infection in a location, or the infection status of individuals in that location, from which you can derive disease risk. And then when it comes to the environment, then you need covariates that are available for the locations where you collect the malaria data, but are also available for the locations for which you want to predict the disease. So, and it needs to be available at a meaningful resolution. So, you know, if you're looking at this map here of malaria in Papua New Guinea, I've got data on rainfall, which is available at a high resolution across the entire country of New Guinea, which means that I can estimate rainfall in the locations where I have the samples, but it also means I have that information for the whole country so that I can predict what the risk of malaria is in a given location, given the amount of rainfall that exists in that location. And the same for temperature. And then here with distance to coastline, what I needed for that was I needed a detailed polygon for the coastline of New Guinea, from which I could then derive a distance measure using a tool in the geographical information system. And then I can extract all of that information for each location in order to build the model and then do the spatial prediction.
Thank you. Yeah, that kind of answered the next question. That is Teresa Kent. She has asked, do you incorporate climate change and socioeconomic data in your spatial models to assess future impact?
Yeah, so in terms of socioeconomic data, a lot of surveys collect information, socioeconomic variables. But often those variables are not available for spatial prediction because you may not have that information in non-sampled locations. So if I ask someone, for example, about their occupation, or I ask them about their income in a survey, I can use that information in a model to adjust the other covariates, the environmental covariates, for the purposes of spatial prediction, but I can't use the information on the association with those socioeconomic variables in the spatial prediction itself, because I don't have that information in the for the non-sampled locations. So I don't know what the mean income is of someone in a non-sampled community because I haven't surveyed them. So you can still use that socioeconomic information, but only in order to adjust for the environmental covariates, not as part of the spatial prediction, because you don't have that information on the non-sampled locations. It still can be useful information to have, but it won't assist with the spatial prediction. So it means that the spatial predictions don't incorporate spatial variation in socioeconomic parameters. And so that is a weakness of the approach. I think, did you also ask about climate change?
Yeah.
All right. So climate change is a tricky one, because in order to forecast the impact of climate change on infectious diseases, you need a really long time series so that you can properly characterize interannual dynamics of disease over an extended time frame in order to be able to predict what's going to happen in the future. And often surveillance data sets are not available for a sufficiently long time frame to properly characterize those interannual dynamics, and they might even be interdecadal dynamics. You know, so temporal variation in disease can happen at a daily scale, can happen on a weekly scale, can happen on a interannual scale, on a seasonal scale. It can happen on an interannual scale. It can happen on a decadal scale, and you can't characterize particularly those interannual and decadal scales unless you've got a really long time series. And not only do you need to have the long time series for the disease, you also need to have a long time series for the covariates that you're using. I didn't cover the example in this talk, but I will show you another slide where I did exactly that for another disease, which is not a water-borne disease, but it's a terrestrial disease called echinococcosis and this is some work that we did in China, where we looked at the landscape change over a 30-year period. And we can see that there's been significant landscape modification in Ningxia, which is this region of China. And that's reflected in the land cover classes that we see. This is information derived from satellite remote sensing. And you can see the emergence of the dark green land cover type in the bottom of the map. And we believe this is driving risk of echinococcosis in this region of China. And we were able to obtain a long time series of echinococcosis data from this region of China. And we actually found that there was a signal in this land cover change when it came to looking at the emerging risk of echinococcosis. But in order to do that, you need to have that long time series of environmental data, and you need to have that long time series of disease data. and that's not available for many, many infectious diseases.
Thank you. So the last question is about how you deal with the scale. When you have different data sources and you are trying to understand a transmission cycle at the nexus of vector, virus, human, animal environment interface.
So often the scale of an analysis will be driven by, it'll be driven by the public health measures or the public health decision making that needs to be undertaken. So, for example, if you're working with a national disease control program for malaria or for schistosomiasis, and they want to be able to prioritise the allocation of resources across an entire country, and that there's significant heterogeneity in risk of that disease across an entire country, you may focus on an analysis at a national scale. If you're interested in, say, eliminating malaria from a particular cluster of communities because they're a potential source of infection for other communities around them, and you want to stamp out individual cases of transmission, you might go down to a much more higher resolution. But of course, then you need to have data available at that resolution. So it really depends on the operational questions that you're wanting to answer and the scale at which data are available, and also the particular dynamics of the disease. So with malaria elimination, you're interested in very local dynamics. Schistosomiasis control, you might be interested in a much higher, in the dynamics that exist at a much lower resolution or a much greater scale. So it's no one size fits all. It depends on the question, depends on the operational requirements, depends on the epidemiology of the disease, depends on the availability of data.
Yeah, a related question again, that is the climatic data comes as a continuous data at a specific spatial resolution, while the malaria data is discrete. It's a location point data. How do you interpolate malaria data to align with the climate data to come up with your spatial model?
Yes, so basically what you do is you extract the environmental data at the location where you sample the disease data, if it's malaria, for example. Often what you might do is you might actually put a buffer around the sample location and extract a summary of the environmental data within that buffer, because an individual may be exposed. The area where an individual might be exposed might be bigger than just the location of their household. So if you're thinking about dengue, for example, you might be bitten by a mosquito at home, you might be bitten by a mosquito at work. And so extracting information on the environment in your household may not be relevant to where you're actually exposed. And so you might seek to smooth that out by putting a buffer around the household and assume that someone was exposed within a given distance of that household, and then use that as the measure of the environmental variable. But then, of course, you need to replicate that when you do the spatial prediction. So when you predict risk in a different location, you need to actually put buffers around all of those locations and extract a summary value in that particular location. So you use those smooth surfaces to extract information on the variable in the location where you have the disease data, and then you use it again when you predict, because you need to know what the profile of the environmental conditions are in a prediction location in order to estimate what disease risk will occur there on the basis of the associations that you estimate in your model. And then you also need to do validation, which is something I didn't get into very much in my talk.
Yeah.
Yeah,
sorry.
I'm going to have to go soon, but yes, I'm happy to answer one last question.
Yeah, and the last question is, have you ever worked in Brazil, namely in state of Para? And there is also a question in Portuguese to Milton. I translated it and saw that is INPE involved in the work, because she or he saw you on the panel. So
I have done work in Brazil, not in Paraná. I've done work in Sao Paulo, in Presidente Prudente, on visceral leishmaniasis. I'm currently doing I've just actually spent this morning reviewing a paper by my collaborator there. I'd love to do more work in Brazil. If you're interested, get in touch.
Thank you. So with that, we come to the end of questions. We have covered all the questions. And Milton?
Yes, thank you, Nandini, and thank you, Archie, very much for this wonderful presentation. Very well illustrated with interesting case studies about different diseases associated with water and also in different places. I enjoyed it very much. And I'd like to mention that I already answered to Claudine. Thank you very much for your interest. And I can provide you, it's easy to find my contact, so we can keep talking later on if you have interest. I should also mention that you all can join the ONWARD network, which is an open network for water-associated diseases studies, as Nandini already mentioned. So please do that. And also, I'd like to thank their center, José Moutinho, Dan, and all the colleagues from there for their very efficient cooperation, as always. And also to thank all the audience for coming here today. So the floor is yours, Nandini. Maybe Shubha wants to make a comment.
Thank you very much, Archie. I won't keep you. I see that you're late for your next appointment and we are running late. But thank you very much and food for much thought. And we will be in touch.
Thanks, Shubha. Thanks, Nandini. Thanks, Milton. It's been a real pleasure and an honor. Thank you so much.
Thank you. Thank you for a wonderful lecture. Thank you, Jose. Tanya?